So why Pseudomonas fluorescens? Well, really, we isolated this organism because it was great at making proteins. So this is a very purpose-built platform. It's designed for expression. We didn't take something from the lab and then try and shoehorn it into making proteins. So once we identified this organism, we built a toolbox around that. So applying combinatorial strain engineering, using a multi-omics approach, we've been very focused on how to get this organism, which is a great protein expression organism, to make soluble, active, properly folded protein. And we'll go through some of the features of this toolbox and the organism and why it is, or we feel it's so great. So a little bit about the platform and the toolbox as we've built it. We have a number of off-the-shelf plasmids with typical features, so different promoters, different ribosome binding sites, etc. So we can tune transcription, translation. Another really important feature is our large collection of signal peptides. Pseudomonas fluorescens is a gram-negative organism, so it does form disulfide bonds in the periplasmic space. So we need to get protein out there to make sure that it's properly folded in those cases. And we combine that with an array of different host strains. So these are engineered to help proteins to fold properly, really focused on quality. So we've got a number of folding helper overexpression strains, protease knockouts, things that can reduce nibbling and chewing of your protein of interest. And then more recently, we've been focused on some more advanced tools for non-canonical amino acid incorporation, other tools for improved folding, and then streamlined recovery. A little bit more about P. fluorescens. It is a gram-negative organism. Another really great feature of Pseudomonas is that it's an obligate aerobe. That really helps us to achieve these high cell densities without a lot of the negative effects of going into anaerobic fermentative growth, either at large scale or small scale. It's got a proven safety profile. The other advantage as we go into scale, and I haven't talked too much about our scale-up capabilities, but we do have a number of products that are on the market made using Pseudomonas fluorescens. And the other great advantage is that it gives a really low cost of goods with high titers, a really inexpensive mineral salts media, all animal origin free, antibiotic free. We do have 6 products that are on the market, FDA, EMA, WHO approved, using Pseudomonas fluorescens as the expression platform. We've got a team that's very capable of scale up, process development. We've done well over 25 different tech transfers around the world and worked with a really broad array of different proteins.